Hello, my name is Wolfgang Miesbach from the University Hospital in Frankfurt, and it's my pleasure to give a short overview about the study results of the HOPE-B trial on behalf of the co-authors. The goal of treatment with etranacogene tesapamovec and AAV-based gene therapy in general is to deliver a one-time injection that establishes sustained factor IX activity in the mild to normal range. The HOPE-B trial was the first phase 3 study in hemophilia B and had the largest cohort of gene therapy for hemophilia B to report to date. It is also the only trial to include participants with neutralizing antibodies to AAV5. In addition to patients with pre-existing neutralizing antibodies to AAV5, patients with prior hepatitis B, prior hepatitis C infection, and HIV infection were included, which may enable more patients being treated with gene therapy. Participants were in total 54 adult males with severe or moderately severe hemophilia B who had previously been on continuous prophylaxis for at least 2 months. Participants were evaluated in a lead-in period of 26 weeks to assess bleeding events occurring during standard of care continuous routine factor IX prophylaxis. Patients were dosed at 2 times 10 to 13 genomic copies as a single 1-hour infusion. No prophylactic immunosuppression was required. Participants were then followed up weekly for 12 weeks, then monthly through the first year to continue every 6 months in the long-term follow-up period through year 5. The primary endpoint for the study was comparing the ABR following treatment with the study drug and prophylaxis for non-inferiority between the lead-in phase and the 52 weeks following stable factor IX expression. The factor IX activity of all participants up to 18 months post-treatment was 39% at 6 months and 36.9% at 18 months. The primary endpoint was met, showing a significant reduction in ABR for all bleeds by 64% at 52 weeks steady state compared to lead-in period. The mean ABR during the lead-in period was 4.19 as compared to 1.52 during months 7 until 18 post-treatment. This statistically significant reduction with a p-value of 0.002 not only met the primary endpoint for non-inferiority but also demonstrates clear superiority of etranacogene tesapamovec to prophylaxis in the lead-in period. Reductions of over 70% were seen in spontaneous traumatic and joint bleeds. The number of participants with 0 reported bleeds also increased significantly from 25.9% at lead-in to 63% at 7 until 18 months post-treatment. Most treatment-related adverse events were mild. Transaminase increases were reported. Corticosteroids were required in 9 participants, but the mean duration of corticosteroids, including taper, was only 79 days. Factor IX expression was maintained throughout the study. 1 death was found to be unrelated to study treatment. 1 case of hepatocellular carcinoma, which has been reported in detail previously, was reported, but after detailed analysis, no association to the study was found. Quality of life also was assessed using the Hemochol assessment. Most domains show an improvement in Hemochol score. Significant improvements were seen in 4 key domains: participants' feelings, their work and school life, in their treatment, and in their future domain, with reductions between 6 and nearly 15 points. So finally, the HOPE-B trial was the first phase 3 study in hemophilia B, had the largest cohort of gene therapy for hemophilia B to report to date. It included patients with neutralizing antibodies to AB5 and met the primary endpoint with a statistically significant reduction of ABR. The factor IX activity showed a continuous increase of up to 36.9% at 18 months post-treatment. Thank you very much for your attention.